Last time around, we talked about how DNA, which stores all the information in how to make a human being and an animal and so forth, can be translated into messenger RNA, which then is transcribed into protein, and proteins can do things like make the structure of muscles and act to regulate all the chemical activities in the body. And the Human Genome Project, which was going to sequence the entire length of DNA and succeeded in doing that, was supposed to result in a golden age of medicine right now, but didn't. And there are several reasons for this, and epigenetics, which is what I talked about last time, is one of them, but it is perhaps not the most important reason. We actually don't know for sure yet, because we encountered a lot of complexity where we didn't expect it. So what I'd like to do now is talk about, besides epigenetics, which was dealing with how DNA is folded to allow it to translate into messenger RNA, some other complications that occurred. And one of the complications is that we were looking at a concept called a gene, which I described in the description area. And a gene is a piece of DNA that codes for a functional protein. That's the original definition. So you've got some DNA over here, let's say, and that is translated into messenger RNA and transcribed into protein over here. And that was the deal. But they discovered, before the Human Genome Project began, that there was another type of gene that they didn't really expect. And what happened here is that this process that we see called transcription didn't occur, and we just made RNA, but it wasn't called messenger RNA. Instead, it was just RNA, or it had some prefix here, which I'm not going to go into because there's a bunch of them. And what happened when they sequenced the human genome is, first thing, they were expecting to get initially this many, 100,000 genes in the human body. But almost as soon as they started, that number had been downgraded. And as I read some of the journal articles in different publications, that number kept going down and down. And now the number of genes is going to be estimated at 20,000 or as low as 15,000. And that came as a big surprise because it didn't seem like that would be enough to operate a human being. Those are the instructions. So they were aware that we could redefine the gene not only to deal with this concept of protein, because that's what they were used to. That was part of the central dogma. The central dogma is you start over here with a gene and it's translated and it's transcribed and you got your protein, and the protein is what's used to do the rest. But this RNA, excuse me, over here, they discovered that that stuff was actually being used not to help with the process that we see here, but to do the work of proteins themselves. So this was heresy, because even though we were aware of it occurring, we thought it occurred very rarely. But now what we're finding is that there may be as many as 20,000 genes that code for RNA that does stuff that's not related to making proteins. Now you might say, well, what's the problem? Well, the problem is we've got those all sequenced here in the DNA. We got all that. But all the techniques we have are for studying the genes that comprise stuff that makes proteins. And working with this RNA that's going to be doing all kinds of stuff that we thought just proteins do, except a little bit, maybe we've got these other types. Turns out possibly to be 50% of all genes. And that threw a huge monkey wrench into our predictions for what the Human Genome Project could do. So besides epigenetics, RNA genes, we could call them, was one of the things that wasn't expected and is leading to a delay in the golden age of medicine. Now, another one was an assumption that was made by the vast majority of scientists that when you had a problem, some kind of genetic disorder, that it was caused by something called a common allele. And these would be the type of disorders that lots of folks have. And what happens with this, like for instance, hemophilia is a situation where there's just one gene that's bad, and a form of a gene on a chromosome, which is a linear piece of DNA, is called an allele. So if you have a bad allele, you might be a carrier, and if you have two bad alleles, then you can have the disorder of hemophilia. And so that is an allele, a form of a gene that was pretty common. We're not saying it's a majority, but it's fairly common in what we call the gene pool in the population. And so there's a number of other disorders we discovered like that, and we assumed that basically that's how it is for most diseases, because that's what we're used to. Well, the reason we expected this is that's all we were finding, because we were looking under the lamppost at night where there was light. We found what was easiest to find. But then what happened is, as we started to look at the human genome, we found something that we weren't expecting. And now the majority of scientists who used to think that diseases were caused primarily by common alleles discovered that they were caused by fairly rare ones, and they were rare and they were going to be in combination. And this is what really makes it tough. Here's the basic deal with this. Let's imagine I'm going to draw some alleles here as just like little colors. So I'll say, okay, right over here we're going to have an allele, and I'll use a bunch of colors as an example. So we'll have another one over here, we'll have another one over here, and I'll just kind of do this a few more times so I don't bore you too much. One over here, and then over here we'll put another one. Let's see, I got to use a different color. Okay, like that. Except instead of just having 5 of them here, in truth, you can have a very, very large number of different forms of these alleles, perhaps 100 to 300 of them. And it turns out that you get the disease if you have this allele, and this allele, except not just those 3, maybe 20 of those. But if you don't have this over here, and if you have this over here, then you're fine. And what happens is you get combinations which cause the disorder and combinations which don't. So it's hard to figure out when someone is going to have the disease, because what's happening is these different genes, these different products of genes are interacting with each other to cause the disease to occur. Not as over here what we see as common alleles, but instead as combinations. And even worse, they're rare. And the techniques for studying genes were all designed for common, not for rare. So now we're upscaling our ability to work with rare. So in both cases that I mentioned here, we had a problem not only with the fact that things are far more complicated, but also because our techniques were not sophisticated enough to deal with these things. And that's because things are complex, especially in biological systems that have evolved over eons of time. You get all sorts of unexpected complications. So this initial idea that we had over here, this very basic notion here, which is the standard dogma, is being modified and modified and modified. And to tell you the truth, there's more to the story than just this, but But that's all for now.